So as Ben said, I'm Paramvir Dehal. I'm a scientist at LBL, and I'm the APS lead. And so for today, what we want to do is introduce our new samples framework that we've added to KBase. The purpose of this webinar is just to kind of get the introduction out so that you understand what we're building, why it's useful, why we built it. And then we'll have a series of follow-up webinars that go into a lot more detail of exactly how to add your data to KBase, add your sample information to KBase, link your data and link your analysis to samples, and then how to kind of share all of that and use it in your work. So next slide. What I'm going to do is just explain the basics, the nomenclature, why samples are useful in KBase. Shane Canon is going to explain our samples design, what samples actually mean in KBase, and then the current scope, and then the roadmap of like the new features we'll be adding. And then Pamela Weisenhorn will go through an example analysis that will really show some of the power of what can be done with samples today. And then we'll talk more about some of the future webinars, topics, and then Ben's survey. So next slide. So again, just the speakers. I'm the lead of the platform science group. We do a lot of the R&D and new development for KBase. Shane is my co-lead, and he's also the chief architect of KBase. And Pamela is our subject matter expert. She's a scientist at Argonne National Lab, and she also participates in the ESS Dive program as a community-funded partner. So traditionally in KBase, we've been focused on the raw data. So people kind of come to KBase, they've got their reads, they've got their genome, and then they analyze it. They process it with our apps. And the gap that we have is that it's hard to know, like, where did those reads come from? What was the experiment that they were a part of? Users tended to kind of encode a lot of that information into, like, the file names, the object names, but this isn't like a great solution. So one of the things to really kind of improve the analysis of the data is we have to be able to capture the sample information, where that data came from. So the new part that we're adding is this sort of left-hand side of this figure, where we're trying to capture the sample metadata and some of the preparation metadata. So this means that we'll be able to link different types of measurements together. So you would be able to know that something like the geochemical measurements are linked to a particular set of reads, a particular set of KBase data objects. But then also being able to kind of associate more kind of abstract content, like just properties of, like, what samples, what attributes of the environment is associated with that data object. And so we've been also trying to work with some of our partners at ESS Dive. We're using the Caesar format for our samples, and Shane will get into more detail of that. And then we have a kind of pretty elaborate set of documentation to explain how to use it. If we go to the next slide. So what we're trying to do is capture the sample metadata, which is shown on the left-hand side of this graphic. And this includes all of these sorts of attributes of how that sample was collected, some of the properties of that sample, and then link that to the actual experimental data. So if you have metabolomics, chemical abundance data, metagenomic sequences, or amplicon data, you can link those attributes together. And then all of that sample information is available then to our analysis tools. So if you make a heat map or some other like profile of your data, all of those sample attributes can be used for the clustering, can be used for labeling and filtering as well. So we go to the next slide. So the samples serves as a path for exploration and discovery of data in KBase. And capturing sample data is one of the key things to enable fair data representation, so findable, accessible, interoperable, and reusable. So when you share data, everyone will know what sample it came from, what the sample properties were. We're developing user interfaces so that users will be able to link data and locate the data through search. So if you can do a more complex query, so you're able to find data not just by its name, but by where its geolocation is, or geolocation plus additional information about what data is linked to it. So if you wanted to find subsurface data, subsurface samples that had amplicons and metabolomics data, you'd be able to do that query, get just the samples that have all the data represented, and then you can share that data, and then other researchers will know exactly where that data set came from. So as we kind of progress through our samples development, the kind of power of these linked samples and data will just continue to increase. We're hoping that users will be able to edit the sample metadata and make it more rich as time goes by. So if there's errors in some of the sample attributes, other users will be able to call it out, and we can kind of improve the sample metadata as we kind of progress. And then we want to provide a resource where we just sort of ingest all of the kind of publicly available sample resources, primarily like, samples from NCBI, EBI, and NMDC. And if we go to the next slide. And so now I'm going to hand it off to Shane Canon, and he's going to talk about how the actual implementation was carried out.
Great, thank you. Okay, so I think it's useful to kind of start by making sure we all sort of think about things in a common vernacular. So when we talk about a sample, what we mean is a physical piece of material that's been collected from some volume at a certain time. And then, once we have this as kind of an entity in the system, then we can use that to attach information to it. So we can attach various attributes like the ones Paramvir showed in the diagram about where the sample was collected from. And then we can also use this to link measurements to, and so this allows you to use it both in your analysis, but also it allows you to eventually bring together data from kind of different modalities and be able to do more sophisticated analysis with that data. And then finally, it helps you kind of organize all of your data as well. Okay, so next slide. A little bit about how we're capturing these inside the system. What we actually do is each sample has a set of controlled and user-defined attributes. And so both of these types can be used in an analysis, and you'll see some examples of that in Pamela's slides. But the reason we have these concepts of controlled attributes is those go through additional kind of validation, and they have some more some conceptual meaning that's really attached to them. So that allows us to eventually, use that attribute potentially across the system because it has a common meaning. So, and then user-defined attributes basically go through less validation. They might be consistent within a given sample set, but there's really no guarantees past that, right? To take, your sample data and get it into the system, you use templates. These basically describe how to map from a given sort of column in a spreadsheet into these controlled attributes that we've created in the system. And then from there, you can link raw data and analysis results to the samples. So Paramvir talked a lot about sort of the ambitions we have around samples, and there's a lot that we eventually want to do with it, but this is just kind of the first baby step in being able to deliver those capabilities. So, this is samples 1.0. I'd say, it has enough features to do interesting things, but, it's not going to necessarily be able to solve everything you want to be able to do in samples and analysis today. So we've started out with kind of specific use cases that Pamela talked about a little bit more around subsurface analysis kind of data sets. And we've chosen templates from CSAR, as well as the Enigma project from DOE, as a way to kind of start fleshing out these designs. CSAR is good because that is a public repository you can just submit your samples to. So if you have, those types of samples, maybe they're already registered in that system. It does the validation that I talked about earlier. So it will validate that things are the correct type. It can do range validation. It can also do ontological validation. So for some of the terms, we've said these have to come from the OMVO ontology, and it can validate that those are valid OMVO terms, for example. And then currently today, we can link a couple of basic data types to these sample objects. So you can link reads, amplicons, and chemical abundant measurements today. And we're going to be expanding that in the future. I'll talk a little bit more about our roadmap plans. We have viewers to be able to go and look at that sample information, and also to look at what we call sample sets. So when you load these templates together, stay back on the previous slide for a little bit longer, those become a sample set, and that's kind of your basic unit of kind of analysis that you'll use in the system. And so we have tools for uploading these samples, and also ways to kind of validate that things went well, and also give you feedback when they didn't, so that you can make corrections. And then again, Pamela will show you some of the examples of what you can do with it today from a scientific standpoint. Next slide. So today, what it allows you to do, you can organize your data, you can compile and link metadata and attributes to these that can be used in analysis. You can use some tools to inspect and make sure the data that's been loaded is correct. We've seen this already be useful where it's spotted little mistakes and even some of the test data we've used. It can generate various types of visual elements, including maps. You can manage these so that you can share them with your organization or with colleagues or collaborators, and eventually publish them. And then you can use these to connect, again, various measurement data, and use this to perform analysis. So you'll see the details of that later. Next slide. Okay, and then again, this is just the first step. We're going to be working on being able to link other data types to the analysis. For example, we really want to be able to for people to be able to go from reads all the way through various forms of analysis, and all those products be kind of linked through the samples. So we're working on some of those pieces right now. Another thing we want to be able to do is be able to manage sample sets so that you could create you can subsample those, or create supersets of those from other sample sets to do different types of analysis. We want to, right now, samples is kind of a very flat kind of object. We want to add more hierarchy to it so that you can arrange things in forms of like studies or experiments. There are other systems that do a similar model to that, so we're going to try to borrow some of those concepts. And then we want to, another one we want to do, and this is going to be challenging, is more generalized ways to link in other forms of measurements, traits, and species that are all linked to these samples. We know this is important for a lot of use cases, so we're working through some of the design elements for that as well. And eventually being able to even capture experiments and protocols so that those can be used, captured in the analysis, as well as just provide additional context for the samples. We have support for importing, like I said, CSAR and some kind of custom templates today, but we want to expand that and be able to interoperate and import directly from other sources. So So for example, we're currently working on what it would take to import from biosample. And then eventually all of this is around being able to link data across the system. So what we're really trying to get to is the point where you can bring in your data, you do your analysis, but then you can also find other data in the system based on properties of your samples that are probably relevant to your analysis. Okay, and so now Pamela's gonna talk a little bit about what you can do in the system today, sort of scientifically.
Thanks, Shane. So as Shane mentioned, the initial focus of this effort was on subsurface sample types. So, when we're uploading samples, as we'll go through in more detail in the next webinar, but as we're uploading samples, we fill out a template and upload that template. And so the first set of templates that we used were from Cesar and Enigma. So these were templates that had already been designed that we adopted and then worked to develop the proper validation of the different fields within these templates. So Cesar, as Shane mentioned, is associated with IGSN numbers. And so this is really good for sort of geosamples. So thinking about soils, rocks, sort of geological features. And a lot of the metadata is focused around that at the moment, although Cesar itself is expanding to include more microbial type data in it, as well as we're expanding to look at other templates that can be uploaded. So some of the data fields that are available to capture some of this information are sort of the location. So where on the globe are you, latitude and longitude, as well as where on the globe are you in terms of elevation above sea level or depth into the soil or rock surface. What that material type is. So what is that physical sample made of, and how was it collected? And so that's both in terms of the sample type, was it a core? And then also sort of how was that core collected? Any other sort of collection method details, who collected it, and any sort of other descriptions that you have, including things like biome and other things that have invoterms that we can use to validate the ontology. So what I'm going to present, I'm just going to show you a little bit of what we can do, and I'm going to walk through sort of some of the output of apps from doing looking at amplicons and sort of doing analysis and visualizations based on that sample metadata. I'll have one quick slide looking at sort of the functional predictions of taxa, although you could do this at the community level as well. And with amplicons, the community is collected from the sample level data. And then we can also examine relationships between microbial community composition and environmental factors or other sample metadata that is contained within those templates. And we'll have additional webinars on each of these topics that kind of walk you through the apps and how to use it and the specific details. So I'm really just sort of giving an overview of what it looks like to do this. Right. So one of the things you can do is sort of explore the data by looking at the sample metadata. So this is a collection of samples that I took through time at 4 different wetland locations. And so here I have divided up my OTU analysis by those sample locations. So you see sort of the 4 separate entities. And if I wanted to say, look at this differently, I have these different time points, so I could break it up by time point and group all of the different locations together. I could do that as well in the app. I could just select, my sampling time as opposed to looking at the sampling location that I entered. All right, and so this just kind of gives you a quick way to make sense of the data and how samples differ from each other. Similarly, we can do clustering analysis by the sample metadata. So you can do right now NMDS as well as PCA ordinations to see where these samples are clustering, how similar these communities are to each other. You'll notice here that we kind of have two different clumps. We have sort of the orange and blue over here and the red and green over here. And so that tells me, based on what I know about the information I've encoded here in this, because there aren't actually fields for all of these things in that default template yet, that the wetland is more important than the plant community that's dominating that area. So the T and the P here indicate different plant species that are located within the same number, which is the wetland location, right? And so we can kind of just take a quick look. If I wanted to break it out even further, I could, separate this out again. I could do the sampling number or the date or any of the other factors that I have on this data. I can also do basic statistical analysis of the data to say, and this is new in KBase, the ability to kind of do this, and it's because we really need to have an ability to sort of group replicates, and you can do that by looking at samples by various factors. So here we have an ANASIM, or analysis of similarity results, on the top, as well as a Permanova, or permutational multivariate analysis of variance, on the bottom, for those samples that I showed you previously, broken down by the location. So that's both the wetland and the plant species. So that's why there's 4 groups here. We'll see that in both cases, we get a good test statistic and a good p-value. So these are significantly different. Even if there's the test statistic on the ANASIM says there's some overlap, and that's likely due to those plant species showing the overlap in the previous image. Right. We can also do what's known as a SIMPER analysis, and that identifies for us which of those taxa are driving the differences in those samples. So here, the sort of bluish purple and the green that have high bars for both of these different species are one wetland, whereas the small red and small violet purple bars are the other wetland. And so we see that there's very different average abundances between these two different kinds of wetlands, and there's some variation with the plant species for most cases, but the real driver is again that location in terms of these individual taxa that we can identify. We can also use PyCrust or FAPROTAX to predict the function of both the individual taxa as well as communities. And so here I'm showing sort of the for individual taxa. So you'll actually see that these bars are quite small, and so these labels don't capture all of the different OTUs at the moment, but you'll see the heat bar indicates the abundance of an individual metapsych pathway, with the pathways being these individual columns here. And so we can do this, we can make predictions of metapsych pathways, IMG phenotypes, tiger FAMs, PFAMs, KOs, ECs, COGs, and we can do those things both for these individual taxa as well as for the whole community. And so that would be a sort of, again, at that sample level. Some key considerations when organizing this data. So I sort of pointed this out, but to make sure that you capture it, each piece of information in the metadata should be contained in its own column. So here I just had the name of the feature as, this wetland with the number as an identifier, and then the letter on the end for the different plant species that were located at the same spot. Ideally we would have each of these broken down into their own column. So there'd be, all of the numbers here, all of the letters as a separate column. So then we could split that out and look at just plant species, just the wetland, And so it's a really powerful and important thing, especially when considering sort of some of these downstream analyses. It's always possible to sort of regroup things later, but it's much more challenging to split them out. And so when you're creating these initial sort of metadata fields, having more and with a single piece of information in each column is really important. And then also, although it takes a lot more time and effort sometimes, and sometimes only a little bit more time and effort, providing the more detailed information makes this data more fair, right? So instead of having a column for salt treatments and having high, medium, and low values, it's helpful to include the salinity values that would be used for each of the treatments, and always important to keep track of and keep the units. But if you use sort of the exact salinity values, the benefit both to you and other members of KBase after you have shared your data is that it allows linkage to the other data on the system. And that's one of the most powerful things about having samples on system and about doing these sorts of analyses on system as well, is that you can take advantage of the other shared data on the system. So you can, if you're seeing a really interesting salt effect at a particular salinity value, you can look on system, or you will be able to look on system for other data sets that have sort of similar salinity values to see if they have the same thing. Or if you see a big transition between medium and high in your treatments, you can actually go to what those salinity values are and find samples that maybe come from locations with salinity values in between there, and see if it's sort of a gradual transition, or if there's sort of a critical point that's hit in between those samples. That can help you to provide context and a deeper interpretation of your data and improve your science. And this goes both ways, right? So you'll be able to use other people's data to do that. And if you share your data on system, other people will also be able to take advantage of your data in that way. And we see this also as a potential way to identify new collaborators that may be outside of your network, or not somebody whose work you're familiar with, but maybe you have similar samples coming up. And so this can help you to maybe engage with them and be like, okay, well, I see these really big differences, and I see this in your data, and we have these sort of data sets that aren't exactly the same, but that we can do something together. Is it possible for us to ask questions that are sort of at the interface between what we have? So with that, I will hand it back for the wrap up.
Yeah. All right. Thank you, Pamela. Go ahead and go to the next slide. First, just a reminder that this is, again, just the first kind of overview webinar that we're doing around samples. And so future ones that we will be presenting on is the next one will be sample creation and management. So this really will get into details of like, how do you prepare your spreadsheets to upload? How do you work through validation errors, validate that everything looks correct, and manage updates? So really getting into the details of things. Next will be samples and amplicon analysis. Pamela covered a lot of sort of what you can do in her part today. In this one, we'll actually go into the details of like how to do that analysis yourself, including uploading your amplicon data and getting it linked to your sample metadata. Next will be samples and chemical abundance analysis. So similar to what was done with the amplicon analysis, but looking at the chemical abundance data. And then last will be showing how you can link these together and do even more sophisticated types of analysis. What was that? Okay, go to the next slide. I think Ben's been posting the link to the survey, but please do fill it out. What this is really useful for, in fact, I want to get to some of the questions that were asked, but we're really looking for feedback on our prioritizations. And so we have things that we think are important, but if we hear from you, then it can help us adjust our plans, and we might move some things around in our kind of schedule based on what we're hearing from the users. So being able to understand things like what type of templates you are using, where you're currently storing your data and sharing it with others, what types of analysis you want to be able to do that's linked to samples, what other types of measurement data you're collecting that you want to be able to link to that. These are all useful things for us to know so that we can properly, prioritize things. So please do fill out the survey. And I think that was it. I think the next slide is just question. Oh, wait, acknowledgements. So, this is, there's been a large team of people that have worked on building up this capability, both software developers, UX developers, our subject matter experts that really help guide our implementation, the outreach team that's helped with preparing the documentation, as well as the webinars. Ben's picture should also be up here. Apologies for not adding that in as well. And then, of course, also the PIs, so Adam Ark and Chris Henry and Bob Cottingham for their leadership. So I just wanted to make sure to thank all the people that have contributed to this. And with that, I think we can take any questions. I'll go through some. There's really, okay, there has been a few extras asked. I'm going to go ahead and answer one, but Paramvir, if you want to jump in and add anything more on it, please do. So one question was about, like, how do you, make sure you're in the right format so that you avoid, different words for the same thing. Or another mistake could happen is using the same word for something that's conceptually different. And the example that was given was like glucose. So we will talk about chemical measurement, chemical abundance data and samples in a future webinar. I think that's very relevant to this question. But in general, the templates are how we're trying to make some sense of the chaos. And so putting them into the correct template helps us know that this is what you mean it to represent. And we've defined a vocabulary for these controlled terms that also comes into play. But then for measurement data, there's the various specific data objects, and we try to make sure that those can properly represent the data as well. So we'll see more of that in the future, but that's a high-level answer. One comment about usability, and we do appreciate this. We do try to factor that in, but it's always good to be reminded of that, and we'll definitely consider that. Is there any specific guidance on limbs like metadata? I think I would say, Paramvir, what would you add here?
Yeah, well, my comment there was like in the future, we're going to have we're going to expand samples to include experiments and experiment information so that users can link protocols and link samples together as part of a single experiment or a single study. Right now, you can encode a lot of the experiment information as part of the user-defined fields in most of our templates. It's depending on what labels you use, it's harder for other users to interpret that, but you would still be able to kind of filter on some of your limbs-like information within the system if that was something you needed to do. But we don't have specific guidance right now for what the best practices are.
Yeah, it's definitely a challenging problem. I think it's going to take us a while to kind of work through some of the design elements for that. Another question about, can you use the microbiome data analysis comparing two groups, experimental groups and control groups, for example, using sequence from 16S? And so, yes, that is supported. I think one thing that we still need to have additional support for, and we're still working through, is some aspects of things like traits, for example, and some of this could also get into the experimental design as well. So for now, the best option is to just use things like user-defined fields in the samples, probably to capture some of that, but we're hoping to have more sophisticated models for that in the future. I don't know, Paramvir, anything else you would add there?
Yeah, I mean, I think in the future, there's also like more on the microbiome, and maybe we'll have a future webinar on this, of like being able to link microbiome data and like metabolomics data. So there, you are comparing specific samples together in order to, kind of understand the treatment versus control. And a lot of that is kind of done right now based on the attributes of the sample. So it would kind of have to be encoded at upload time. The other aspect that we'll talk about in the future webinar is that effectively the unit of the analysis are these sample sets. So you can decide which samples to put into any particular sample set. So if you want to partition your data in different ways, and then run the analysis tools there, it's just a matter of searching, filtering, and constructing your sample set, and then running the analysis. So there's a lot of different ways to help slice your data and do the right comparisons that you want to do.
Right. Okay, so I think that was the last question that I saw in the Google Doc. But please, if you think of something afterwards, you can still add to that. We'll check on it for the next couple of days just to see if there's anything to expand on. And again, fill out the survey, and please attend some of the future webinars, especially if they kind of align with what you're thinking about trying to do with samples. Certainly the first one, I think, is kind of a must, because it's going to really help you do the very first steps of getting your samples into the system. And then the ones after that are going to be a little more topical. Yeah. All right, Ben, any other closing remarks?
None for me. I think this was a really helpful overview, and yeah, I just would encourage folks to look out for the announcements about upcoming webinars and to fill out the sample survey and the beta testers form. Especially if you have data, or you are thinking about designing your experiments right now, and you're sort of trying to map out how you're going to do your analysis. That makes you a great candidate to work with us to think about structuring data. So and what kind of data you need to get. So please, indicate that in this survey. And yeah, we can work with you one-on-one to help you get your data in the system and get things working nicely. So yes, thanks to everybody for coming out today. Like I said, you can follow us on Twitter, on YouTube, to see recordings of this webinar and other webinars. And of course, please sign up for a KBase account. That'll put you in the loop for all of our communications about new features, trainings, conferences, new papers, and that kind of stuff. So thanks everybody for tuning in today, and we'll see you in a few weeks.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.